Good afternoon, and thank you for joining us for today's webinar entitled Embracing the New R&D Paradigm with Structured Data and Insights. Today's session is presented by Benchling and Astrix Technology Group. Next slide, please. Before we get into today's session, which is going to combine presentation materials as well as demonstration of live software, I want to tell you a little bit about Astrix Technology Group, the sponsor of this webcast series. This is a webcast series that's running through November. We've invited industry experts to come on and share their thoughts on the state of technology in lab informatics. And we're very happy to have Benchling, a partner that we love working with us today, and they're going to share their insights and even some really interesting demonstration with their product. But briefly about Astrix Technology Group, so you know who we are. If you haven't been on any of our webinars or in any of our webcast series yet, we're an informatics professional services and staffing company dedicated 100% to serving the scientific community. The company was established in 1995. It is privately held, but it originated out of the IT division of a company called AgBio, which at the time was about $300 million life science research organization. Today, Astrix operates from 8 offices with over 650 employees and has its headquarters in beautiful Red Bank, New Jersey. We predominantly offer two major services, or we have two major divisions. The staffing division, which offers private and public staffing of scientific and IT resources. And on the professional services side, it's focused on supporting science-based organizations with traditional, lab informatics implementation and consulting services. The customers that we work with are Fortune 1000 life science enterprise, government, research organizations, large and fast-growing IT staffing and compliance needs. The mission has and always will be to deliver scalable, sustainable solutions in IT and staffing for the scientific community. On the professional services side of the fence, what we're delivering is business process analysis, enterprise architecture design, laboratory technology selection services, development and implementation, computer systems validation. So we work with solutions like a Benchlink, for example. We can come in and kind of help a company architect the solution, figure out how these solutions are going to work within their environment, make sure that it is configured and customized to meet the specific needs of your laboratory. And then we do the post-implementation work, like the training, computer systems validation. And then there's also the ability for us to even potentially staff individuals in your organizations to maintain and manage these systems, or of course do other non-technology laboratory services as well. At the end of the day, we evaluate, implement, and manage systems to enhance the collection and processing of scientific data. So that's enough about Astrix. Let's go ahead and introduce our wonderful speakers for today. We have Rashi Takar as a product marketing manager at Benchling. So she has a bachelor's degree and a master's in immunology from McGill University, and previously worked at Forte Bio as a senior product manager for the Octet systems. Prior to that, she worked as a scientific researcher at Genentech in the research organization where she developed immunoassays to support IND filings. So very pleased to have her. And Nalini Yadav is a scientific solutions consultant at Benchling. She received a master's in biotech from Fort Valley State University and a PhD in pharmaceutical science from the University of Hawaii at Hilo. I hope I pronounced that right. And prior to graduate school, she worked in the information technology field, designing and implementing and maintaining a small business network. So there's a lot of brain power on the call with us today. So I want to go ahead and turn it over to our speakers, and I hope everybody enjoys the presentation.
Thank you, Kevin. Great. Good morning, everyone. Good afternoon. Today I wanted to share Benchling's vision and how we're helping life sciences companies embrace the new R&D paradigm with structured data and insights and really innovate faster than ever before. So just briefly, I'll be going over some of the company background and also the solution that we offer. And then Nalini will come and show you what the Benchling demonstration, really go into the platform and demonstrate kind of the power of Benchling. And then the last 10 minutes we'll spend on Q&A. So if you have any questions, as Kevin mentioned, please add it to the chat. Perfect. So Benchling's mission has been to accelerate life sciences for the betterment of humanity. Benchling was started back in 2012 by a team of scientists from MIT. They saw how software was changing every other industry and were frustrated with what they had available for their own science. So the company's vision is to build software solutions that could truly accelerate how life sciences R&D is conducted. We're primarily sustained by a growing customer base We also have support of our original investors like Andreessen Horowitz and Benchmark. These are the same firms that back end companies like Instagram and Twitter. So there's a shared vision of what software can do to truly transform an industry. We're able to invest heavily in our product development, which means new capabilities coming out every single week. And we can invest in our customer success with dedicated support teams and value engineering to really help our customers get the most value from our solutions. Currently, we're about at 250 employees, and we have more than 350 customers of leading R&D innovators across the life sciences. So the biopharma industry has evolved tremendously over the past decade, and we're proud to work with hundreds of some of these leading companies that have developed new advances in medicine and discovered new therapeutic modalities. Some of these include Pfizer, the Regenerons, Gileads, and also Sorrento and Mammoth. This is clearly not an exhaustive list, but we wanted to highlight some of our key customers. We also support customers in the AgBio and industrial biospace, where some really innovative technology is being developed to improve food supply and environment sustainability. We're proud to work with hundreds of these leading companies that have developed new advances such as Bolt Threads, Ginkgo Bioworks, and Clara Foods, to name a few. So now delving into this, modern life science R&D requires truly a new way of working or a new R&D paradigm to stay competitive, which has become especially important during these pressing COVID times. To stay competitive in this industry, companies are focused on building scientific platforms that look beyond individual discoveries and focus a platform that spans multiple modalities, such as adoptive cell therapies, antisense oligonucleotides, and hybrid modalities like antibody drug conjugates. And these can be evolved for continual innovation. As these platforms are getting more and more sophisticated, they're building specialized teams to truly focus in on critical areas of development. This requires more orchestration between teams with more data flowing back and forth. And finally, life sciences companies have to be able to collect and bring together data from a complex matrix of resources. And now they're amassing data at unprecedented scale. The challenge is not just about the volume of data, it's about the organization and the structure to this data. In many companies, most of this data remains siloed and not analyzed in the broader context. It's key to be able to collate the relevant data quickly to drive intelligent decisions at every level. Most of our customers share with us pain points they have on their existing solutions with their legacy silo technology, whether that's paper-based, spreadsheets, homegrown solutions, or siloed point solutions. We hear continuously from scientists how frustrating it can be to always be entering data manually into different systems that don't talk to each other, then spend time trying to piece it back together for review presentations. This not only wastes time, but it also impedes the flow of innovation. R&D leaders also struggle to have the data they need to make timely and data-driven decisions. They have often become dependent on others to integrate and compile data from multiple sources, which can lead to delays in reporting or missing data resources. And the sheer number of applications used and the siloed nature of many applications makes it very costly and inefficient for IT teams to maintain. They're often using costly resources to maintain software instead of investing these resources in more impactful data science projects. It's clear that a new kind of digital solution is needed to help overcome these obstacles. And that's where Benchlink comes in. Benchlink's Life Sciences R&D Cloud is designed to give you that 360-degree view of your R&D innovation. Benchlink's applications span the R&D process from experimental design, in silico biology, sample tracking, and process management. There are 7 applications that Benchlink offers: molecular biology, notebook, registry, inventory, requests, workflows, and insights. The key differentiator that Benchling brings is that all of these applications are offered from a single interface. This makes it easy for scientists to leverage what they need without actually leaving the software or having to enter it multiple times. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. This makes it possible to query the system for sample and process intelligence from sequence level all the way to the final product batch. Benchling is built on a modern cloud infrastructure fully managed by Benchling and offers state-of-the-art security and control, so you never have to worry about that. And most companies have a rich ecosystem of instruments and software applications and data stores in their R&D. So Benchling provides an extensible data model with a full set of REST APIs and out-of-box connectors to move data into and out of the system. So let's take a quick look at how Benchling can help support your R&D transformation. So Benchling brings the ideal traits that any digital solution should have in R&D. And there are 4 core attributes to consider, which we feel Benchling truly excels at. 1, it's been built for complex science. It models the full range of molecules and biological entities you focus on, which means having both the data model built fit for purpose. And we'll get a quick glimpse of this when Nalini does the demo. Benchling can also adapt to your process and support the processes and workflows that your team performs on a completely native SaaS platform that can scale, iterate, and evolve over time and is always up to date. Benchling also enables key decisions because it empowers users with data at every level, giving scientists insights about their experiments and team leaders data on their programs so you can make both scientific as well as operational decisions quickly with complete and accurate data. Benchling is also easy and intuitive to use and ensures that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will help keep them compliant and consistent with the rest of the R&D organization. Let's dive deeper into how Benchling enables each of these core attributes, and Nalini will follow up with a demonstration of these attributes in the Benchling platform. So Benchling is built for the complex science your company works on. We'll help you find the right way to model your data during implementation, and from there, you'll be able to connect your experimental results back to your samples. We're able to model anything in Benchling. You can model any type of biological or chemical entity, from DNA and amino acid sequences to cell lines, viral vectors, to antibodies and proteins. The model can be easily evolved over time by your admins as your science evolves and as you add new programs. This also provides end-to-end sample traceability. When you have the right data model for your science, it helps connect every part of the process together. You can see the relationship between sequences, the plasmids that the sequences were put in, the viral vectors that were generated, the cell lines, the proteins, and the batches and the passages of each lot. You can quickly follow this lineage anywhere in the platform without having to guess where it came from. You can see the experimental data that is associated with every entity and start to truly automatically compute certain fields that will save time from having to do this manually. All of this serves as the foundation for a system you can use to accelerate your R&D innovation. Once you have the right model in place, you can then start building a process on top of this model. Life sciences companies each have their own unique approach to their science. We can adapt Benchling to any number of life sciences products. We can organize and optimize your R&D processes at each step. This means we can get you up and running quickly with a configuration over customization approach that reflects your science and gives the power to your IT teams and administrators to help evolve the system to your needs by adding additional fields as they need without spending weeks and months having to recode. Also increase process visibility. Benchling can ensure the data and experiments are always captured in a standardized manner that improves data quality and integrity at every step. We also have embedded visualization and process intelligence to get insights throughout the process and not just at the very end, without having to manually collate data from multiple sources. Once you have the right model and process management, you can then have the right foundation to pull key insights from your system. So the third core attribute is enabling truly like data-driven decisions. So no matter what your applications are, modern life sciences companies need to access their data quickly and with context around it, where it comes from. Benchling offers this level of embedded analytics across all applications through its insights. Benchling provides actionable insights at every level with insights. So you can drive scientific and operational decision-making with data coming from within experiments, across specialized teams, and across entire divisions. Data can be used to optimize experiments, understand program resources, and uncover team and departmental bottlenecks. So if you see on the left, in this example, you might be looking at a plot of our data for virus titer yield. If you want to understand this data better in context, say for example, as a new scientist or a new team member comes on board, you can click right into this virus entity link and see all the associated metadata with it. And even click into its components, such as the transgene plasmid sequence and its associated annotations. This is all just a few clicks and a very powerful way to understand the science in context and act quickly. As powerful as it can be to have a centralized solution for R&D, we know that this only works when scientists and other stakeholders use the system as designed and feel that they can use the system. This is where an easy and an intuitive interface truly comes into play. Most R&D software applications in use today were designed decades ago, and the user interface falls way short of what most of us expect. Additionally, R&D teams often find themselves switching back and forth across apps or docs to enter and find data, causing a lot of lost productivity. Benchling's intuitive interface was built around the idea of a single interface. So no matter which applications your team decide to use, they will always access it from the same entry point. This saves time having to switch between applications and makes it easier for users to access data and collaborate with colleagues. Time-saving features are embedded throughout the platform, such as a very powerful search filters and ability to drop in links to entities using the @mention key. Benchling was also designed for productivity. So in addition to the ease of use of the interface, Benchling was designed around productivity. It helps automate repeated tasks and minimize the amount of manual data entry required from users. All of this helps your teams to be more productive, work faster, and improves the overall adoption of the platform. So Benchling is truly designed to work with your existing ecosystem. We understand most of these companies you have hardware, you have different software and business functions already implemented. Benchling provides that digital hub that truly integrates your R&D ecosystem. We know how important it is for data to flow freely into and out of Benchling to support key R&D functions like IP filings or IND submissions. Our customers routinely take advantage of data import and export capabilities that exist throughout the platform. But it's also important to connect directly to other critical lab systems. So Benchling has a robust set of APIs that facilitate connections to other lab software. We also have a Benchling for Lab Automation product that provides framework and out-of-box connectors to liquid handling, as well as different analytical instruments. And our data warehouse can be made accessible to move data into your existing business intelligence tools, such as Tableau or Spotfire. All of this is designed to help our customers design and accelerate their R&D digital transformation. Now let's take a closer look at Benchling's architecture to see how all of this comes together. So Benchling supports a wide range of R&D processes with applications that span the entire workflow, from design to experimentation to sample traceability and analysis. But each of these applications resides in a unified interface. All of these applications are accessed by scientists from a single point of entry through a common web browser, and Nalini will be showing you that as well. When we implement Benchling for our customers, our experienced professionals take time to understand your science and recommend the most appropriate data model that captures every step of your process and your products, from DNA constructs, vectors, to cell lines, to proteins, to reagents and animal models. All of this can be configured by our team without any code, and they can quickly prototype so you can ensure it will work as you want before it goes live. Having a unified data model is critical to be able to ensure full traceability and truly provide that chain of custody for your samples. All of the Benchling applications are built and delivered through the Benchling platform, which consists of an infrastructure that delivers security, scalability, and integration capabilities. We publish a full set of REST APIs to connect the software in and out of your to connect the software and data systems your labs have in place. There's a data warehouse that I mentioned, and also you can connect to third-party BI tools. Because of the sensitive nature of data flowing through this architecture, security is a top priority for Benchling, and it's thought about in every step of our design process and across every step of this architecture. So Benchling really has put security at the forefront of everything we do. We support hundreds of enterprises where data security and control is mission critical, including top global biopharma companies and public and private institutions like the Department of Defense. We think about security at every level, from how we plan out our business to how we train our engineers and into our infrastructure and application level security. Our security model is designed to protect our customers against internal and external threats and make sure that we can be trusted guardians of their core IP. We strive to be transparent in all of these processes so every customer can feel confident in knowing how Benchling manages and protects your data. Before I pass on to Nalini for a demo, I wanted to kind of show you a quick case study about Rubius Therapeutics. Rubius Therapeutics is a clinical phase company in Cambridge and uses a novel cellular engineering platform that engineers red blood cells into cellular medicines. Prior to using Benchling, they operated from a variety of manual systems, such as paper, Excel, some homegrown systems that were very difficult for their scientists to collaborate around. It was also a challenge for them to build reliable processes on these and access data for reporting purposes. They adopted the entire Benchling platform, which includes the 7 applications, back in 2017, and have expanded it to include the entire Benchling platform for their scientists. Their team can now collaborate better around a shared view of their data, and their project management is now centralized in a way that has accelerated and optimized project delivery in a significant way. This is just one of many examples we can share of how Benchling customers can accelerate their R&D using Benchling, and we're happy to share others, or you can read more on our website. Now I'm going to pass it over to Nalini to kind of walk through the actual platform and give you a sneak peek into Benchling.
Okay, thank you, Rashi. Can everybody see my screen?
Yes, the screen's coming through great.
Great, thank you so much. So, as mentioned before, Benchling is a cloud-based platform which you can access via your browser. Here I'm using Google Chrome. And after logging into Benchling, you will open up to this home screen. Now, if you notice here at the top, if I expand the calendar view, this is where it's actually linked to the experiments in your notebook entries. This is the kind of high-level or project management view for your individual scientist or lab manager. And here you can see various different notebook entries, the days and dates on which they span, and they're color-coded by project. So if we go back to the actual notebook entry itself, so this is a notebook entry that was actually created from a notebook entry template. So you can templatize your entries to consistently capture your data across all of your different scientists for various different experiments that you perform over and over again. So some things of note here is that at the top we have some standard text editing options. Over here to the right, there's some options that you can toggle on and off in your view. And at the top here, we have some tabs. So we have the Notes tab, which is where your actual notebook entry lives. And then you can also attach protocols to your notebook entries in Benchling. And those can be, any file type that you like, whether they're PDFs or Word docs, etc. You can attach them directly into Benchling, and they would be attached to this particular notebook entry. You can also attach files within the body of the notebook entry itself. You can either drag and drop from your computer, or you can actually insert as attachments here as well. Another feature that's pretty neat in Benchling that, Rashi mentioned earlier is @mentioning. So let's say I wanted to ask my colleague a question, and I wanted them to take a look at this particular notebook entry at this particular spot in the notebook entry. I simply use the @ symbol and type in a minimum of 3 characters, and Benchling brings up a pick list of various different things that I can mention. This is the user I want to mention. I can just click that, and then it selects them. And then Benchling will actually send that user an email saying that you've been mentioned in this particular notebook entry with a clickable link to take them directly into this notebook entry. So it allows for very easy communication and collaboration in this way. So now let's take a little look further down here. So as mentioned, Benchling has multiple ways in which you can add structured data to the system. And one of those ways is by using these functional tables. So the first type of functional table we have here is a registration table. And this is going to add items to the registry or the structured database, as it's called. So what we have here is a table that's automatically set up based upon the type of entity, in this case a cell line. And the cell line has been configured so that it is required to collect certain metadata fields. And those metadata fields correspond to the column headers here on this particular table. So you can actually hand type in information if you would like, or you can actually upload from a spreadsheet. And I actually have already done that here. And then once you click submit, Benchling actually assigns a unique ID to each of these items that are then placed into the structured database. Let's say then we have our cell lines, we've added them to the database, and now we want to put them into the inventory or sample tracking system. So for that, we would then use, again, from within the notebook, we would utilize another type of functional table. And functional tables, as I didn't mention before, are tables that actually perform some sort of function within Benchling's solution system. So here we have a transfer table so that we can actually put items directly into the inventory or sample tracking system from, again, within the notebook. So we have this table set up. I could actually, rather than typing or copying and pasting information from the table above, I could simply click add samples and it would pull that information down for me. I would simply choose which table from this particular notebook entry I wanted to pull from, and then I could add those to the table. I select my location. So again, we're having the user connect directly to the inventory or sample tracking system from within the notebook and seamlessly, so they don't have to think about the fact that they are actually going into the inventory system. So let's take a look at some of these items. So again, these are clickable links. So if I wanted to take a look at this particular cell line that was just created, I could click on this and it takes me directly into the registry or structured database. So here we have the cell line name, the registry ID, which is a unique ID assigned to the item as soon as it is put into the database. And then we have the schema here, which is the cell line schema or how it's configured. And then we have the specific metadata fields that were set up or configured for all cell line schemas. On the right-hand side, we have over here a description tab where you can enter in any text information, any notes. You can attach files such as certificate of analyses if you would like. And then we have some additional tabs. So again, we are now looking at the registry or structured database. And from that, we can click on the storage tab to connect directly to the inventory or sample tracking from within here to see exactly where this cell line is located, in what cryo vials, and where those locations are in our actual physical laboratory, including information about the current volume. We can also go to the results tab, which I'll show you in a little while, how you can aggregate your data that's being collected against your different entities. And then we have a data model system that can be set up to not only link different entity types together, but to, as Rashi mentioned, to set up data models where you can have lineages where you have parent and child entities. So I've set up a parent entity as a cell line and a child entities as engineered cell lines. So from within this particular cell line entity in the registry, I can then click on this tab and see all engineered cell lines that have been made from this particular cell line. So let's go back to our notebook entry now. So we've seen how the registry is connected, and we see how that structured data is collected there. Now let's take a look at how the structured data is collected as far as the inventory. So let's click on one of these cryovials that were just created that we put one of these cell lines into. So here we have the barcode. So the unique ID is also the barcode of the item in Benchling's inventories tracking system. And you can actually connect your printer directly to Benchling system, so you can print out your barcoded labels from within Benchling itself. And then here we have basic information about what the contents of this particular cryovial are. We can also see the actual hierarchy of where this cryovial lives. It lives in this particular box. And if I click on this, it navigates me to the box view. So this is a schematic drawing of the box and all the items it contains. I can also go further back out to the rack and the actual liquid nitrogen tank that contains this as well. And over here on the left preview panel now, we see the actual inventory application. And if I were to expand this caret, it actually takes me to a dashboard view. So this is where I can actually query the inventory or sample tracking system using the user interface itself. Okay. So now if we go back again to our notebook entry, another type of table that's very useful in Benchling system is a result table. And this is how you're going to actually collect your experimental data, not just your metadata, to aggregate that onto your entities. So here we have a protein production result table where I'm actually collecting protein being produced by these engineered cell lines at various different time points. So if I were to then actually click on this engineered cell line, I can now go to the results tab and I can see all of the results that have been collected against this engineered cell line, collated and aggregated onto this particular engineered cell line entity, and as well as have clickable links to the actual notebook entry where that information was collected. And then if we actually go into our Insights application to pull all of this together, so let's say we wanted to know which engineered cell lines are producing the most amount of protein. Well, we can actually utilize Benchling Insights, where you can write simple SQL queries to query the data that's now been collected in these structured databases and output in also a table form as well as a visual format such as this line graph here. And if you notice, the most recent data has not been reflected here yet, so I just simply refresh the dashboard, and now we will see that this new engineered cell line 025 has been added to this as well. And as Rashi mentioned, from this view, I can actually click on this and directly go into and see the metadata for that particular engineered cell line. And if I were to click on this particular transfer plasmid, it would then take me into the actual sequence for this DNA sequence, where I then now have access to all of my molecular biology suite of tools. And then finally, one last thing I wanted to show you was, first of all, the Insights application can also be used to query and keep note of different stocks in your inventory system. So if you have metadata collecting date information as far as expiration, et cetera, you can set parameters and see exactly which ones are expiring within a certain date range, as well as you can keep track of certain volumes. And you can see volumes of your various different types of entities. For instance, I'm looking at an engineered cell line here. I could look at various different other types of entities, and I can see which particular entities I'm low on in volume as well. And then finally, I keep saying finally, but actually one more thing I wanted to show you is that, as mentioned before, we do have Benchling for lab automation that you can utilize to connect to various different instrumentation in your laboratory, such as liquid handlers or analytical instruments. And you can utilize a run object. So this is a run object that's actually embedded into a notebook entry. And here's where you can actually connect Benchling's items in the structured database, such as items in your inventory, such as these particular 96 well plates, as well as any entities in your, and you can actually utilize Benchling to create instructions for your liquid handler or analytical instruments, which you can then either manually take over to the instrument to give it its method or instructions for the robot for what it needs to do to perform the action, or you can actually utilize the robust set of API endpoints that Rashi mentioned also to be able to connect that directly. And then the output file can then be ingested back in Benchling and processed to connect back to those items in the structured database as well. And then finally, the last thing I wanted to show you was just how easy it is to configure Benchling. Everything is point and click. So if I wanted to create a new schema for a new type of entity, let's say I wanted a mouse type of entity, and I wanted to just point and click and create this, I can simply do that. I can add my metadata fields. So I can, let's say I wanted to connect to a particular animal model. There's different types of fields you can choose for collecting information on your metadata here. And you can link to other entities as well. And this is where you would set that parent link here. And of course, if you wanted to edit your entities, you could simply click into them, and it's again point and click to edit them as well. And that is all that I have. So I will gladly turn it back over to Rashi now.
Thanks, Nalini. Perfect. All right, I think we'll open the floor now for questions, Kevin.
Great, thank you. Yeah, and thank you for a wonderful presentation and a great demo. What I'm going to do is I'm going to go ahead and stop the recording right now. We typically